Hi, everyone, and welcome to Global Genes Facebook Live on genetic testing. We're so excited to introduce you to an opportunity to receive an Exome Next Trio test and to our partners over at Ambry Genetics. So my name is Meredith Cagle. I'm the Senior Director of Patient Engagement here at Global Genes. And Global Genes is a nonprofit advocacy organization who works to connect, empower, and inspire the rare disease community. We've been around for 10 years. In fact, we're celebrating our 10 years this year. In honor of our Global Genes 10th anniversary, Ambry Genetics very generously donated 10 Exome Next Trio tests for us to make available to individuals in the rare disease community. If you were in attendance at our summit or watched the summit via our live stream, you may have heard us announce that back at the beginning of October. And so the Exome Next Trio test includes whole exome sequencing of the trio, which is of the patient of interest plus 2 first-degree relatives who are usually the biological parents. And it uses next generation sequencing methods targeting the 20,000 nuclear genes. So before I introduce you to Kelly, who is the Director of Clinical Genomics at Ambry Genetics, and she's going to tell you more about genetic testing and the Exome Next test, I do want to let you know that the application form to apply for an opportunity to be selected to receive one of the 10 exome tests is available online now, and you can access that link through our Facebook posts or by going to our website at globalgenes.org. It'll be available through Sunday, December 9th at 11:59 Pacific Standard Time. We will be selecting, randomly selecting from the individuals who submit an application and notifying recipients on December 10th. This test, due to the testing itself, is only available to individuals if you live in the United States or Canada. This Facebook Live session will be recorded and available for continued viewing after our session is done here. We'll be answering questions throughout it, so please do submit them for myself, for Kelly, and we'll do our best to answer them during the session. If we don't get to the questions or we need to get some more information, we will look into them afterwards and we'll continue to answer them. If you're watching this recorded and you're asking questions, we will still continue through December 9th responding to those questions. So with that, it's my pleasure to introduce Kelly Hagman. Kelly received her BA in psychobiology from Scripps College at the Claremont Colleges and an MS in genetic counseling from the University of Arizona. Kelly joined Ambry in 2009 as a director of genomic services, and in 2011, she was chosen to lead the launch of the very first commercially offered diagnostic exome sequencing test on the market. Kelly has seen the evolution of exome sequencing from the days it was only offered to researchers to the widespread popularity today. She has more than 40 peer-reviewed publications in molecular genetics and genomics, most focusing on exome sequencing. And we're so excited to have you here today.
Thank you so much. All right, so again, thank you to Global Genes for the opportunity to speak today about Ambry and what genetic testing is in general, and about exome sequencing specifically. I'd like to provide everyone with a little bit of background information, what the genetic testing process looks like, what you can expect when you're going through the process, and what the results look like, and how it could potentially impact medical management and care for you or your child. I'll start by giving a little bit of introduction to Ambry Genetics. I also want to give a little warning that I have a little cold I'm getting over, and I'm 6 months pregnant, so a little short of breath, so I might talk a little slowly. But I'll start by talking a little bit about Ambry Genetics. Ambry is a genetic testing company that is devoted to providing answers to patients who are seeking genetic diagnosis. Ambry focuses on 2 main areas. One is predisposition testing for patients that are looking for a potential predisposition to a genetic disease in adulthood, most notably like cancer predispositions. But we also have a large focus of our testing on patients with rare diseases. And the goal there, and that's the area that I work in, the goal there is to provide a diagnosis, and we do everything that we can to try to provide a diagnosis for everybody that sends in a sample to our laboratory. And I'll talk a little bit about how we really try to go above and beyond to provide a diagnosis for everybody, and that is our main mission, is to identify a genetic diagnosis for everyone. So a little bit of background: what is genetic testing in general? I'm sure everyone has a basic idea, but basically it's a test that examines changes in a patient's DNA. We refer to these changes as mutations or alterations. And these are alterations that lead to disease. So there is variation in our DNA. Obviously, that's what makes us different, what makes us each unique, but in terms of looks, personality, different abilities, and so on and so forth. But there are certain areas of the genome that are the same in everyone, and they need to be the same in everyone because they perform very basic functions. And when those are mutated or altered, that can lead to disease. And those are what we're actually looking for, those really rare changes that are in the really important areas of the genome when we do genetic testing. And why is it important to do genetic testing? So a lot of times the people that in pediatric clinics, for example, pediatric genetics clinics, patients that go there, about 10 to 15% of them will receive a clinical diagnosis. So a clinician will actually look at the patient, identify all the symptoms, maybe take a look at their unique features, and can provide a clinical diagnosis. But that's only about 10 to 15% of patients. And the rest of them they don't know what was the underlying cause of their disease. So that's where genetic testing comes in. And once a genetic diagnosis is established, it's important for patients because it can actually guide clinical management. So we have an idea, once we know the gene that's altered and the symptoms that are generally associated when a particular gene is altered, we know what to expect. So clinicians are very savvy about knowing what treatments to provide for patients once we know the underlying cause, other medical interventions, referrals to particular specialists. There could be some pre-symptomatic screening for other symptoms that are expected to arrive later. Also, anticipatory guidance and support group referrals, appropriate educational planning, advocacy groups, things like that. Also, establishing a molecular diagnosis will allow you to know how it was inherited in the family, how it came about, and how it could happen again. So that it provides family planning. So you can do carrier testing and also potential prenatal diagnosis. Also, understanding that knowing what the underlying cause is will actually lead to additional research. So even if and a lot of times with rare diseases, we find it all the time, there are patients that will have a mutation in a gene, and they may be the only patient that ever had a mutation in a particular gene. So these are very rare findings. But once you find that, it allows for research. So we can find, and Ambry has collaborations with thousands of researchers all over the world, ones that generally specialize in very rare genes and rare diseases. And once we find a mutation in a gene, we try to hook up with those researchers to further characterize that gene, and it can help the family know what to expect down the line. Also, probably the biggest one, the one that patients find the most helpful, is it provides an answer to why. So patients often want to know, or often feel like it was their fault. So I think a lot of times when you have a genetic diagnosis, it alleviates that. It had nothing to do with anything that the parents did. It was something that just happened in our DNA. And mutations happen in our DNA all the time. It's a part of evolution. It's a part of, it's actually healthy to have mutations all the time, but sometimes they happen in genes that are critical to particular basic functions and lead to particular conditions. Also, exome sequencing has kind of been known as the test that can, quote, end the diagnostic odyssey. And that's actually recently come under fire a little bit because people feel like, well, once you have the diagnosis, it's not the end of your odyssey. It's sort of the end of one chapter and the beginning of the next. But it does at least answer the question why. And you can then go on to do your research to determine what happens next. But it can end the time-consuming, often very invasive, and often very costly process that it takes to get up to a diagnosis. So when we first launched Exome back in 2011, we had patients that had been undergoing multiple different genetic tests for, a decade. One of my favorite cases was two brothers that we tested that were in their 20s and their whole lives had been getting different tests as they became available. And we were finally able to find out what was going on with them by doing exome sequencing. There were two brothers, and there was also a sister in the family who was unaffected. The family assumed, and the clinicians assumed, it was probably X-linked, so affecting boys only. And we turned out it was actually not a autosomal recessive disease, so something that we learned that was new. And the sister was able to do carrier testing and find out that, she was not a carrier and we didn't have to worry about passing it on to her children. And when we talk about exome in general, basically, so we're talking about sequencing of roughly all the 20,000 genes in our genome. But before exome sequencing was available, in pediatric clinics, using all the traditional tests that were available would provide a diagnosis for about 1/3 of the patients. So that would include tests like, you may have heard of chromosome analysis that can look like microarray or karyotype. That could provide, that's looking at really big changes in our genome. And that could provide a diagnosis for about 10 to 20% of patients that came into clinics. And then also when you add in all the single gene tests, so testing gene by gene by gene, you have to kind of have an idea about where to start when you go that path, and it can be very expensive. Even a single gene test can sometimes be very expensive. And so through single gene tests and then even gene panels that have up to, between 5 to 10, up to 200 genes, could actually end up being pretty costly and time consuming. But adding all of those traditional technologies together only got you up to about 1/3. So 1/3 of patients received a genetic diagnosis. Once exome sequencing came on the market, that doubled. So now 2/3 of patients that go to pediatrics clinics could receive a diagnosis through exome sequencing. So it's actually a very powerful test. And also, it's also so it's about a half to a quarter of the cost of the traditional testing trajectory. So for those people that actually went through testing, starting with like karyotyping and all the traditional genetic testing that were available, went through those first and then went to exome sequencing. Even when you account for those that were diagnosed along the way, overall, if you go straight to exome, it saves about, it only costs about a quarter to a half of the total cost, as it would if you go through all the battery of tests that were available. So about 2 to 3 times more likely to find a diagnosis and about half to a quarter of the cost overall. So obviously that's why it became so popular so rapidly. Also, I think for patients that individuals that we're actually going through a donation of 10 exomes to 10 different families here, but if anyone that doesn't get granted the opportunity to go through this, we also offer a full pre-authorization to go through your insurance, so that could happen without even having to worry about collecting samples or anything like that. So you would basically just send all your clinical information to the laboratory and they'll do a pre-authorization and let you know whether it would be covered by insurance companies. And then another real benefit to exome sequencing is that you're sequencing all the genes in the genome. And today, we actually only understand what about a half to a third of the genes in our genomes do and what diseases they're involved with. So we're still learning all the time. And actually there's on average about one every other day, there's a new peer-reviewed publication that comes out describing a new gene-disease relationship. And so we're learning about these all the time. So when you get your exome sequence tested, we're able to analyze up to that date everything we know about. So even if a new gene disease discovery was what happened yesterday, that gene will now be included in our analysis when we analyze your sample. On top of that, patients that receive, even if a patient receives a negative result, we're still continually reanalyzing. So every time a new paper comes out that describes a new gene-disease relationship, we go back to all of our patients that we've ever tested to see if they had an alteration in one of those genes. And if they did, we'll go and analyze to see whether it correlates with the patient's symptoms, and we'll send an unsolicited reclassification report. And that happens up to 15% of the time of our negative patients. So it's very powerful. So it's sort of like once you send your sample in, as I mentioned earlier, we're devoted to finding the answer. So we'll continually work to, the biggest reason for reclassification is because of these new gene disease discoveries. But we will continually try to find the answer for patients. We also do that through like our research collaborations, as I mentioned before, that we have with researchers across the world to try to further understand what's going on with patients. In general, overall, patients have about a 25% chance to find a very clear positive diagnosis through exome sequencing. If you've never had any other genetic testing before, that rate goes up a little bit. So it can get up to about 50% or so. But in general, there's still, if you do exome sequencing, there's just to have clear expectations, there's still, 50 to 75% of patients that initially get a negative result. Again, they still have a chance to find an answer later in the future, but it isn't, it's not 100%. Not everyone will have an answer identified. We do, as was mentioned, we do trio testing. What that means is we test a patient, usually a child, and two other individuals, first-degree relatives, usually the mom and the dad. If one of the parents isn't available, we can take a sibling or any other first-degree relative. We can also take grandparents as well sometimes. And that allows us, because when we sequence a patient's exome, we find all sorts of alterations, right? I mean, there's actually 200 to 300,000 alterations that are identified. How do we narrow that down to the one that's leading to disease? If we have the parents to reference against, and for example, let's say parents are both healthy, and the patient is the only one affected in the family, we can rule out all the alterations that were inherited from the parents, and it narrows down the number a lot. So that's the reason why we ask for family member samples, understanding that it does take additional coordination with caretakers and with families try to get blood samples from additional relatives. Also, I think, so the testing takes about 6 to 8 weeks in our laboratory. Generally, like if this is we're offering free testing here, so that would be about as long as it would take. If you're doing insurance, as I mentioned before, that can tag on a couple extra weeks because we don't start anything until the insurance has been pre-approved. And that actually does take for exome sequencing, the insurance companies tend to take a lot longer than they do with any other tests that we do in our laboratory. Also, the types of results that can be identified. So I mentioned about 25% roughly detection rate or positive rate. Those are findings that we know that they're either positive or very likely positive, that are very likely to be the cause of the patient's disease. And then we also have uncertain findings, and I'm sure if anyone knows anything about genetic testing, you've heard about VUSs, or variants of uncertain significance. That can be identified in exome as well. So what that means, and uncertain results can happen for multiple reasons when it comes to exome sequencing. One, because we might find an alteration or a mutation or a change in the DNA, and based on the evidence that we have available to us today, we don't know for certain if this is causing disease or not. Those still get reported in the report. It still are provided in the report because we want patients to have as much information as they can along their path. Everyone hates getting those results, but it does happen about 10% of the time. You'll get an uncertain finding. In addition to not knowing for certain whether the alteration could be leading to disease, sometimes we're uncertain whether the gene that we identified is actually associated with that patient's symptoms. So sometimes it looks like the symptoms overlap quite well with what the patient has, but oftentimes it could be, yes, like there's 20 cardinal symptoms of a particular gene, and this patient has 10 of them, but they're kind of more common symptoms. So we aren't 100% certain, so we will actually call that uncertain on the report, whether it's actually that could be the causative finding for that particular patient. Again, we are continually reanalyzing these cases, so that even the uncertain results, we're still always going back and looking at them to see whether if there's any new evidence that has been published or that is available to us, any new data in animal models or any other functional data in the literature that we can add to our understanding of the alteration for those uncertain findings. And then again, I mentioned before, so we still end up having a good bulk of samples that, or patient samples that end up being negative. So meaning we didn't find anything for that particular patient. However, as I mentioned before, I'll just reiterate again that we do continually reanalyze those patients' samples all the time. So whenever new gene disease discoveries come out, there was actually a paper that was put out by the NIH maybe about 5 or 6 years ago, saying that they believed that all of our gene disease associations, or all of our genes that lead to diseases, would be understood by the year 2020. We're getting very close to that, so I don't know if we're going to quite make it, but it does give you an idea about the pace that we're going at understanding all of our genes and our genome to date. Also, I think, so once there is, and I mentioned before, like when you do have a positive finding, you would take that result to your healthcare provider, and then, as I mentioned before, they would be able to provide tailored management for you around that positive finding. So they would be able to let you know, like, whether there are particular treatments that would be available, particular therapies, particular specialists that should be referred to, and also be able to provide to you a likely trajectory for the disease based on past patients that have had similar diseases. So that's kind of a big picture that I have about exome sequencing and genetic testing in general. I'll go through a couple of your questions now. I saw a bunch of questions pop up. So one is, can you talk about the importance of rare disease patients needing to secure clinical or CLIA testing rather than enrolling in an academic study that will often take up to a year for results in a diagnosis? This is a great question. So in general, going through a clinical lab, most providers will not act on or provide medical management for a test that was done in a research lab. So even if you did find something in a research laboratory, they would want to take that result to a CLIA or clinical laboratory to be confirmed, to make sure that they can actually show that it is actually the finding. And then also, which is fine. So I mean, if you do have an alteration that was identified in a research lab, that it's a lot cheaper to just confirm that one particular alteration in a clinical laboratory. However, a lot of times, depending on the research project, sometimes patients are not even allowed to see their results, depending on the nature of the particular research project. But it's very common for patients to not even be able to see their results. That's obviously something that would be disclosed up front when you get enrolled into a research project. And obviously in a CLIA or clinical laboratory or CAP certified laboratory, we have, those labs will have the most up-to-date state-of-the-art equipment, a lot of QC, so quality control measures to make sure that the findings are accurate. And obviously there's a very streamlined process, so it can get done much more quickly. And also, in general, I think, research studies I think is a really great avenue if patients can get into them to find an answer. But again, I do believe that they should always be confirmed in a laboratory. And as I mentioned before, most in a clinical laboratory, and most clinicians will not provide medical management based on those results until they're confirmed in a clinical laboratory. And another question was if the sequencing will provide an answer for patients with autism. So that's a great question. Autism is a hard one. So, autism is kind of a more common disease in our society. So in general, when we're talking about genetics, the more common the disease, the more difficult it is to diagnose. So if it's a really rare disease, we can be much more confident when we find a mutation or a finding in a particular gene. We can be much more confident that that's leading to this rare disease. Generally, they're happening in genes that are very, very important. So we know they're leading to particular findings. Autism is, and there's a whole spectrum. Autism spectrum disorder can be difficult. There are some genes that are clearly associated with autism in a very, very small percentage of cases. So we're talking between 1% and 5% of patients. Also, autism is complicated because it's also a symptom of many other conditions. So we see autism as one symptom of many, many conditions that we get. So we'll have patients that will have many features, including dysmorphology or even epilepsy and other things that could also come with autism. Yes, sometimes it's also sometimes questioned whether the autism, whether those types of autistic symptoms happened due to other events or if it happened due to the underlying genetic cause. So patients that have just autism and no other symptoms, very, very few of them will ever get diagnosed, will have to date have been diagnosed through exome sequencing. If they have autism and other symptoms as well, it's much more likely to find a diagnosis. So yeah, it's definitely a difficult one to diagnose, and I understand it's common enough when we get that question all the time, and we do see samples all the time for families with autism, but it's much more difficult to find the answer if they just have pure autism. And then another question was, can this test identify spontaneous or de novo mutations? You guys have the really good lingo down. Yes, it can. So, and that's a great question. So in general, that's the main reason why it's very valuable to have parents when we do the testing. So most of the time, most of what we find when we do exome sequencing, because we're dealing with children who are very severely affected, and they have parents that are unaffected. So that means most of our findings are a new mutation or a spontaneous mutation that occurred sometime, during the development of that child, or sometime in the either in the sperm or the egg. And so that's our most common finding among all of our findings. So yes. Okay. And then how many of the known diagnoses are treatable? So that's a hard question, I think. So definitely, I think, and there's a range of what's considered treatable. I think so. There definitely are treatments available for probably virtually every condition. There are treatments available. Whether there's a cure, no, right? There's that's much more difficult to identify. There are very few conditions that have like an actual cure. We're getting there with things like CRISPR that we're all hearing about in the news. And I think in my personal view, I think within 10, 15 years, we're going to be a lot closer to seeing that in action to help patients. I'm super excited about it personally. But in terms of treatments, there are certain conditions that have much better treatment efficacy. So epilepsy, for example, are ones where knowing the underlying gene mutation is very helpful because it can help guide treatment to know which drug can be particularly helpful for particular patients based on the gene finding. And then, however, the vast majority, if you go to genetics clinics, they will have some sort of treatment option for every patient. The efficacy of them and the value of them really does range, from depending on the disease in question. How many of them, I don't know if I can put a number on it. A clinical geneticist would definitely be able to answer that probably better. All right. So thank you everyone for the really great questions. We really appreciate it and hope this was informative.
Great. Thank you so much. I learned so much from that, and I hope it was useful for everyone watching. Just as a reminder, if you're interested in applying for an opportunity to receive one of the 10 donated tests that Ambry Genetics has given us, please do complete an online application before Sunday, December 9th. The link is on Facebook, or you can go to our globalgenes.org website for more information. If you're watching this later, please do ask questions. We'll continue to answer them online throughout the next 10 days, and we'll be notifying recipients who are going to receive one of the tests on December 10th. Thank you guys so much.